Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall talk about necroptosis. In the previous videos, we saw how cell signaling through various receptors can either lead to the pro-survival, pro-inflammatory pathway or to extrinsic apoptosis. And if extrinsic apoptosis doesn't happen, then the cell can undergo cell death by necroptosis. So for example, with the TNF-R1 cell signaling pathway, we saw how the cell can undergo extrinsic apoptosis because it uses its caspase 8 to activate the executioner caspases. However, if the caspase 8 is inhibited, and we know a lot of proteins that can inhibit caspase 8, including FLIP that is released by different viruses, as well as cells. Then if caspase 8 is inactivated or is inhibited, now the cell still has another way to die, and that is through necroptosis. So notice that these proteins here, RIPK1, RIPK3, they play an important role in necroptosis. We see the same thing with the FASR receptor. Once again, if the caspase 8 is inactivated or inhibited, apoptosis doesn't happen. However, once again, RIPK1, RIPK3 can be recruited and the cell can go through necroptosis. We saw the same thing with TLR3, for example. Once again, if caspase 8 is inhibited, then the cell can use these proteins and undergo necroptosis. So let's look at the necroptosis story. What happens in necroptosis? So we saw this part, the ligand binding to receptor, and then the recruitment of various proteins. We saw this in many different pathways that we discussed in previous videos. This causes the recruitment of RIPK1 and RIPK3. So these proteins, receptor interacting protein kinase 1 and 3. And this RIPK1 and RIPK3 have these domains called the RIM domains. So they will recruit, they will interact through the RIM domains to give you this RIPK1, RIPK3 proteins bound to the receptor. Most often in the next step, the RIPK1 will phosphorylate and activate RIPK3. The activated RIPK3 will then recruit another protein called mixed lineage kinase domain-like protein, or MLKL. So now MLKL gets recruited towards the receptor site. The RIPK3 will phosphorylate and change the shape of MLKL. So you see the MLKL shape has changed and you see this phosphorylated MLKL. This change in shape or conformation of MLKL causes it to self-assemble to form a large complex, an oligomer. We see this oligomer of MLKL being formed. These oligomers will travel from the cytoplasm towards the plasma membrane as well as towards membranes of other organelles. So we see the MLKL oligomer gets embedded into the plasma membrane as well as into the membranes of other organelles such as the mitochondria. This embedding of this MLKL oligomers makes the plasma membrane more permeable. Also true for other organelle membranes, but important thing to note here is now the plasma membrane, because of these proteins in the plasma membrane, makes it more permeable. And because of this pores, the increased permeability, sodium as well as water will now enter the cell. The cell as well as all the organelles will swell. There will be further membrane damage due to this swelling, and that is going to release all kinds of cellular contents, including danger-associated molecular patterns from the cell towards the outside. And that is going to cause cell death, and these dams will also trigger inflammation. So in summary, in necroptosis, there is plasma membrane disruption. The plasma membrane basically will break, it will lyse. There is cell organelle swelling, release of cellular content because the plasma membrane is permeable or eventually breaks. So a lot of cellular content is released. There is no chromatin condensation as what we saw in apoptosis. And because of this release of all the cellular contents, it's going to trigger inflammation. And that is unlike apoptosis, where there is no inflammation, at least at the beginning stage of apoptosis. Now, if the cell were to go through apoptosis, remember that caspase 8 in that case would be active. And caspase 8 will break these essential proteins that are required for necroptosis. So that's how apoptosis will actually prevent necroptosis from happening, because caspase 8 will destroy these essential proteins necessary for necroptosis. So this is how cells die by necroptosis. In the next video, we'll talk about another cell death pathway called pyroptosis. Until the next video, once again, thank you very much for watching and supporting this channel. Have a wonderful day. We'll see you in the next video.